The rapid emergence of new strains of SARS-CoV-2 virus can potentially threaten the protection offered by COVID-19 vaccines. To overcome this, researchers at the Indian Institute of Science, IISc Bangalore, have developed a new approach that provides an alternative mechanism to render viruses like SARS-CoV-2 inactive. We reached out to the team of researchers who have developed this new class of artificial peptides or mini-proteins that can not only block virus entry into our cells but also clump virions, which are virus particles, together, reducing their ability to infect. In an exclusive conversation with PBNS, Professor Jayanta Chatterjee from Molecular Biophysics Unit in IISc explains to us what are mini-proteins and how do they prevent viral infections or diseases.
See, mini-proteins are basically, as the name suggests, they are basically proteins. Now, proteins are primarily composed of amino acids, and mini proteins, they are sort of an upcoming area where, as the definition goes, that proteins that are composed of less than 100 amino acids could be classified as mini proteins. Now, and the other interesting feature of these molecules is that they have a very compact structure, right? Now, so the compact structure and the small size allows one to sort of decorate their surfaces with any functional groups which can then be used to target your protein of interest. In our case, it was the spike protein of the SARS-CoV-2, but you can take any other target of interest. It could be in oncology, it could be diabetes, whatever. Okay. Now, so in essence, they are a completely new class of molecules which are certainly not antibodies and not small molecules. So they sort of fill the gap between the very large class, which are antibodies, and the very small class, which are the small molecules. So these are mini proteins, and then there are certain advantages of mini proteins. For example, antibodies, they're brilliant, right? There's so many antibodies in the market that are used as therapeutics. But antibodies, there's a complex, production procedure. Then you have stability issues. You can't just store these antibodies at room temperature. And then, the other thing with antibodies is, antibodies, since they're large in size, they do not really like to penetrate tissues extremely well to reach their site of action, if it's necessary. Sometimes, in most cases, it's not necessary. But if it's necessary, then there is a problem. But these small-sized mini proteins, they can do that very effectively. They can happily sort of squeeze their way out into these tissues and then go to the site of action. And since they're small in size, they can be very easily manufactured. Bacteria can actually manufacture, or yeast cells can actually manufacture these mini proteins. You can also manufacture these mini proteins chemically. So that's what we have done in our laboratory. We manufacture these mini proteins chemically. Now, when you are capable of manufacturing something chemically, you can incorporate amino acids that are not typically found in nature. Now, when you are capable of doing that, what you're doing is you have additional, let's say, additional handle to alter their structure, make them more stable, because typically proteins are the way nature is doing it. The proteins are produced and the proteins are degraded, right? So there is a, there's a production and there is a degradation, because that's why, that's what we want. We want reversibility in the system. But sometimes if this is not necessary, if you want the mini proteins to stay in our body in the circulation for a longer time, what you would want is, you would want them to be proteolytically stable. You don't want enzymes to chop them off, right? So chemically, this can be done. You can incorporate unnatural things which can fool the proteases, the enzymes. So the mini protein is very stable. It can stay in your circulation for a longer time. And this is important for the therapeutics, because therapeutics, you have to achieve certain pharmacological properties.
The most recent development by the IISc research team has identified mini proteins that can prevent COVID-19 infections. Professor Jayanta tells us the mechanisms behind it.
Yeah, so there is a bit of a historical background which is really necessary for the viewers or the listeners to understand. So the protein that we designed, the idea was fundamentally taken from a paper published by David Baker's group from University of Washington, Seattle in mid 2020s. So what they did was they are a pioneer in computationally designing proteins. Okay, well, again, mini proteins, let's say. Also, they do design proteins as well. Now, so they reported a work which used the advantage of computers to design mini proteins, and they showed that this mini protein targeted the receptor binding domain of the SARS-CoV-2 spike protein. So essentially, so imagine in a very simple term, SARS-CoV-2 is a spherical particle, and it is decorated by these Y-shaped molecules, which are called as the spike proteins. And the head of those Y-shaped molecules are the ones that essentially bind onto the cell surface. Okay, so they showed that if you can take a tiny protein, they designed it computationally and also experimentally validated it. If you can use that, those tiny proteins can basically go and bind onto those Y-shaped molecules and will inhibit the interaction of this virus on which the spike protein is decorated onto the cell surface. Okay, now that was very interesting. So at that point, for us, because like every other researcher wanted to contribute to the growing knowledge of SARS-CoV-2, we thought that we could also do that. So and at that time, I must say here that also there was a ACRB project proposal call on COVID-19, and my senior colleagues asked me that you could try to submit a proposal and see that if you get funded. Even if you get funded, you could really work in this area for some time. And luckily we got the grant, and that's where all the work started. Because sometimes the grant is absolutely important to do the experiments in the laboratory. If you do not have money, it's very difficult to do experiments. Yeah, so we got lucky, we got the grant, and then we started to work. And the other thing I must say that I had two colleagues in my department, Somnath Datta and Raghavan Varadarajan. So Raghavan has been working with viruses for a long time, and Somnath has expertise in cryo-electron microscopy, structure determination of very large protein complexes. So so we had the perfect background to work in this area. And since the beginning of the pandemic, Raghavan has been working heavily with the SARS-CoV-2. So I thought, okay, perfect. No, I'm in the right place. We could do something. So then so what we did was we looked into the structure what David Baker published and thought that, okay, let us do something. So they have a protein which is a very nice folded structure, and we thought that can we further make it smaller? Okay, because our idea, as I said in the beginning, that our idea was to use a peptide which can circulate in the body for a long time. We could incorporate unnatural amino acids. The one Baker reported did not have any unnatural amino acids, so they would be probably proteolytically cleaved off, and when the molecule is cleaved off, it will not be effective at all. So we thought that, okay, we looked into this, and then while looking into analyzing the structure, we also realized something very peculiar about this molecule. So it turned out that so imagine this is a 3 helix bundle. There are 3 rods, and I can take a pen and probably try to explain you. So the protein was basically 3 rods, okay, 1, 2, and 3. And so let's say this surface is the one which binds to the spike protein, okay, and this surface, this surface is solvent exposed. It does not bind. Then what we thought is, let's do something. We wanted to make this peptide smaller, so what we did was we thought, let's remove this third pen, okay, the third helix. So with this we thought that now if we have these molecules floating in water, because you make this molecules will be millions of those molecules, in fact billions and trillions and zillions, they will be sticking solution in floating in solution. But when they float in solution, since this is sticky, each of these molecules will bind to its partner and form the 4 helix bundle, meaning that now instead so the same thing from the back side, it will bind and form a dimeric structure, okay. And if it forms a dimeric structure, there are 2 of these molecules, but they will stay attached forever, okay. And that's what it turned out, that we saw that so we essentially ended up making dimers Now the beauty of this molecule is, since this part was involved in the binding to the spike protein, now this part would also be involved in binding to the spike protein because it's just a dimer, so it's a backside attached to it. Now we tested that with small proteins, just the receptor binding domain of the spike protein, and it turned out yes, whereas Baker's molecule that they designed was a 1:1 binding, that did not dimerize. The most critical part was that truncation. And also what happens is we did some engineering at this site, because these two cylinders are basically connected by a loop. Now they can go, they can flex, because if these molecules flex, they will not form a very nice compact structure. So we had to do some engineering at this site so that it was retained, and then the sticky patch was exposed, and then it bound. Now, now what it did, so we saw that. Now, okay, so I must tell you one thing. Now let's say that if I do not make this surface sticky, okay, if I make it polar, then the molecule, just two of these cylinders float around. Now when two of these cylinders float around, it will only bind to one of the receptor binding domain. It will not dimerize, because you only have one surface that binds to the receptor binding domain, the spike protein. Okay. Now when we tested the effect of this molecule, we saw that this molecule did neutralize the viruses, meaning prevent the virus from infecting the cells, but it was not very effective. But when we took the dimer of this molecule, just by making that surface sticky, then we saw a almost 50 folds enhancement in the neutralization, meaning that, so because what was happening is, by dimerizing, what it's doing is one of this surfaces binds to one spike protein and the other surface binds to the other spike protein. And by forming this, we showed it by using cryo-electron microscopy and other biophysical experiments as well, it forms a very stable complex. The complex does not dissociate. But when one of this molecule binds to the spike protein, there is a dissociation and association, dissociation and association. When it dissociates, the spike protein is free to infect the cell. Now, if you form a very tight sandwich complex, right? So this guy just does not dissociate. Since it just cannot dissociate, you do not have certain population of that which is free to infect the cell, and that's why we got a very high effect or a very high neutralization effect of this kind of molecules. So that is the most exciting part of our research. So there are steps involved when a virus infects the host cell. So the first thing is the binding of the virus onto the host cell. Okay, and then there are some rearrangements onto the spike protein, and then a fusion machinery. Let's say in a very simple terms, the virus essentially fuses with our host membrane and goes inside. Now, if you develop something that prevents the virus to attach the host cell, then you can completely prevent the virus from infecting. It will probably be in the circulation, but it will not infect. So that's what antibodies do. So they develop, the antibodies essentially go and block the viruses at those spike proteins so that they just don't go and attach. Now these mini proteins basically do the same things. So imagine the virus is coming on, this is my cell, imagine the virus is coming and trying to attach. These mini proteins, they go and they immediately, if they're present in the circulation, they immediately will go and attach onto the spike proteins and prevent the access of these spike proteins to attach the to attach the host cell. Okay, this is our cell. Now, in addition to attaching, what we see, what these guys are also doing is, since dimeric molecules, so these two molecules go and bind. So we think that another virus which is also there in the vicinity, it comes and forms clumps. So one virus attaches to other virus instead of this virus attaching onto the host cell. So this is, and that's how you form probably tiny clusters of viruses, as opposed to the virus essentially going and infecting the host cell. So this is how these mini proteins act. That's what we think. As you can imagine, now this molecule binds to the spike protein because it just does not do it randomly, unspecifically. There is certain specificity which is present in this molecule because of which it binds. So it's like, so there are certain ridges and there are certain grooves which basically goes and fits. Now, in simple terms, what happens is this virus is very smart. So it realizes that, I'm being killed in simple terms. So it starts mutating its regions so that, the molecules don't bind. Now the problem is, just like the antibodies that we have in the market or the vaccines that we have in the market, they are becoming sort of inactive against the Omicron and the upcoming variants. These molecules will also be very inactive against those upcoming variants because, as I said, that this has been decorated to bind a specific virus, specific groups in a specific strain of virus. So if the virus itself mutates and alters its residues so that the groups has changed, it will not bind. So you have to re-engineer this molecule. Yeah, the other thing, one benefit could be that you could reasonably fast engineer this kind of molecules to target that, but for that you have to have the structural information of the virus available, where the mutations have undergone, how the surface potential, surface charge, surface grooves and patches have changed. So that is the problem that we have now with viruses. But if let's say that if we use this kind of molecules, so we are very excited with this finding because it's not just because of the SARS-CoV-2, because we have come up with a novel class of mini proteins that can dimerize their target. So as I was explaining that, since it does not dissociate, so let's say that, you target a protein which is responsible for angiogenesis. Now, a blood vessel growth in cancer, because that is also important, tumor progression. So you have to block angiogenesis, the growth of blood vessels. And there, those proteins, let's say that they don't mutate. If you use this class of molecules to target those, you will form very, very stable complexes, and then they can show their very interesting pharmacological properties. They will be very, very effective for example, in the area of oncology. So because, so as I was saying, that we are excited about the mechanism, we are excited about the novel class of molecules. SARS was just an example to demonstrate that you can actually develop against SARS. Now, if there are upcoming variants for which structure is known, you can also tune this class to specifically target that SARS. So it depends upon the therapeutic intervention. See, for example, in the SARS-CoV-2, since it's primarily in the airways, so through lungs, in these experiments, what we did was, so the molecule was given through intranasal administration. Okay, just like, so sometimes, you have probably seen you have seen patients, they take so who are asthmatic, they take aerosol sort of formulation that directly goes to the lungs. So that's what we wanted. In this case, what we did was we directly, sort of delivered the molecule through the nostril so that it directly goes to the lungs and prevents. And it has to engage the target. If it does not go and engage the target, it's of no use. So now it depends upon where the molecules need to go. So if it's in the circulation, okay, if it through the circulation, if the molecule can engage its target, go to its target, then you can deliver these molecules through intravenous injection. Let's say that, if your target is in the gastrointestinal tract, then you could probably give these molecules orally. Okay. Now, the other problem that I say that, for as of now, we have not assessed the stability of these molecules against degradation by the enzymes. But, we can very easily design the molecules to be proteolytically resistant, because these molecules are primarily designed by peptide synthesis, chemical peptide synthesis. So, yeah, so this is in terms of the delivery.
Will mini proteins work like vaccines? what would be the possible lasting effect or period of its efficacy once injected or consumed in the body?
The vaccines work in a very different mechanism. Okay, so the body essentially, let's say the body eventually develops those antibodies, and it sort of remembers whenever you have this infection, the body starts producing these antibodies and targets and neutralizes the viruses. These mini proteins could be classified as the standard drugs. For example, paracetamol, the standard painkiller that we take. Now, paracetamol, if you have fever, you go to the doctor. Doctor says that take this paracetamol, once in every 8 hours. Okay, so so these molecules, the duration of action of these molecules will depend upon the pharmacokinetics, how long these molecules are circulating in the body. Okay, so if the half-life of these molecules is very short, then you have to frequently administer the molecule. But if the half-life of these molecules is very long, then you do not have to frequently administer the molecule. For example, there are several antibodies which are not vaccines, and in certain cancers, those antibodies are given, and the antibodies, they can go in the circulation for weeks, for several weeks altogether. So you do not have to deliver the antibody. You deliver the antibody, maybe after 1 month you have to go and take another shot in the clinic, right? So it all depends upon the pharmacology, how long this molecule is going in the body. Unlike vaccines, as I said, that the body eventually produces once you prime the body with the vaccine.
What is the prime significance of mini proteins in our biological processes? Do they enhance individuals' immunity?
So this mini protein, now something interesting about this mini proteins are, so nowadays there are reports coming up where scientists are finding mini proteins that are produced by the body itself. Okay, now our mini protein does not come into that category, although it's a mini protein. By definition, it's a mini protein, but it's not what is being produced by the body. Okay, now why is it that we did not know about these mini proteins earlier? Because we, as simple as that, we just did not look for those mini proteins. They are here. They have an evolutionary role. They have a role in our immunity, probably. They have a role in muscle contraction. They have a role in many processes. We just don't know as of yet. Research is being done now. There are few reports where people have found the mini proteins, and they have very interesting roles in several processes. So probably in the next 5 years or 10 years, we probably have a much clearer idea that what sort of roles do these mini proteins play in so in your immunity, in our well-being and our health, and probably also related to your cognitive behavior, everything. It's just that we did not look for those. Now our mini protein certainly does not, as I said, that does not come into that category because it's lab made. It's as because we never really looked into the our genome to find out whether the genome encodes for these tiny proteins. We did not find those, but there are few reports which indicates that there are mini proteins which are encoded by the genome, and something which is again encoded by the genome can eventually affect the genome as well, because these molecules can bind to your DNA, these molecules can bind to your RNA, and it's sort of a feedback loop. So it produces some proteins, and these proteins can finally alter or let's say that it can tune the production of additional proteins. Yes, the answer is it certainly seems to, as of now, it seems to have a role, but I think research is ongoing, not in our lab, many other laboratories, to find out how do they affect the genome.
The study published by IISc mentions that with minor modifications and peptide engineering, this lab-made mini-protein could inhibit other protein-protein interactions as well. Professor Jayanta tells us what does that mean and how will it be beneficial.
So as I said in the beginning, that we are very thrilled to have find this novel class of molecules which can actually dimerize their targets and form this very stable complexes. So now, since this is a very general mechanism, now this could be exploited to any other protein of interest. And there are many proteins, as I said, that are involved in many therapeutic or many diseases. So as I say, one example, as I took that, that in angiogenesis there is a protein called as vascular endothelial growth factor. So that protein is promoting angiogenesis, and people have shown that if you block this protein, you can block the angiogenesis and block the tumor progression. And there are many others. So our lab has currently started looking into those class of proteins in completely different therapeutic areas, where so what we are going to do is basically, now we have a very stable structure. This is a dimeric structure. So what we are going to do is, we are not going to disrupt the structure, but as I said that this surface and this surface has amino acids which basically interact with the target protein. So what we are going to do is use that as a scaffold, use our molecule as a scaffold, engineer the residues that are present on this site. Then you can design basically keys that fits to some other lock. So that's the whole idea, because eventually they are going to again dimerize and block the action of those proteins. And we are also currently interested in taking these molecules inside cells and target, there are many interesting target inside cells, and antibodies, they fail to do so because they are so large, they do not actually go inside our cells to target protein-protein interactions, which are sometimes, many times, in fact, responsible for certain diseases, several diseases, in fact.
Okay, so Professor Jayanta, right now I'm just thinking out loud. You
Yeah,
mentioned cells. You mentioned cells. So, and you also mentioned mini proteins, as in they prevent viral infections and other things. So, once you develop this, can they also actually target one of the biggest disease that incurable disease that we face today are cancers, multiple cancers. They are incurable. So once you can actually find a way to inject it into the cells, can it perhaps help people to cure the cancers also?
this is what we think. Yes, this is. I mean, cure cancer, we don't know, because cancer is a very complex procedure. But what we definitely think that, and that's why we are working in this area, that can we at least induce tumor regression, sort of block the tumor growth, because people have shown that it's possible with antibodies. Using antibodies, you can provide benefit to cancer patients. So, and as I said that, so our molecules are only different from antibodies in the fact that they are smaller in size, but they bind to their target with very high affinities, as antibodies would bind. In addition to that, they dimerize this, which antibodies don't. So that means that a very small amount of our molecule can probably be very efficacious in vivo. That's what we think. Now, to provide a yes or no answer, I think research will tell us, and we are certainly working in this area.
Thank you for watching. Stay tuned with PBNS for more such stories on medical breakthroughs in India.